Thank you. I'd like to thank the organizers for the opportunity to come and speak at this very prestigious institution. I had the great good fortune to visit this city one time before, where I got to go and see the Minar, which is a very, very impressive artifact. But what I'd like to do today is tell you just a little bit about some of our studies where we've tried to understand a little bit about how the system, the cancer as a system functions, and how cells acquire resistance or begin to acquire resistance to endocrine therapies. And for those of you who are not familiar with breast cancer, these are the data from the United States. There's about 200,000 or so new breast cancers diagnosed in the United States every year, and 70% of those express the estrogen receptor. And molecularly targeting the estrogen receptor was probably the first molecular-directed therapy for any cancer, and that was the drug tamoxifen, which we've had since the 1970s. The problem is about 50% of these breast cancers don't respond, or when they respond, they recur later. And that's a problem because that means about 35% of all breast cancers are ER positive and not responding to treatment. And that's comparable to all the triple-negative breast cancers and all the HER2 breast cancers added together. So although people tend to think of the estrogen receptor-positive breast cancers as having a good overall prognosis, and generally that's true, when treatment fails, it still kills a very large proportion of women. There's something to be learned from looking at the annual recurrence rates for breast cancer and looking simply at the differences between estrogen receptor positive and estrogen receptor negative. Now, you'll notice that the patterns are broadly similar whether you're looking at in-breast recurrences, distant recurrences, or mortality rates. So what I'd like to draw your attention to is that the estrogen receptor positive breast cancers tend to kill women very quickly within the first 3 to 5 years. And after that, the risk of recurrence drops dramatically. It's very different for estrogen receptor positive breast cancers. These women their risk of dying increases for the first 3 to 5 years. And that's usually during treatment, because we will give these endocrine therapies for 5 to 8 years. And then there's really no change. It continues fairly high. So that ultimately, after you get to about 5 years, for women that have estrogen receptor positive breast cancer, their annual risk of recurrence continues to increase. And that's not true to the same degree for estrogen receptor negative breast cancers. So there's something different between ER positive and ER negative, and there's something different between what happens here during treatment, those breast cancers that recur on therapy, and those that recur many years later. So we tend to think of these as being classical resistance because the cancer is coming back during treatment. And this tends to represent the re-emergence from dormancy. So the cancer seems to go to sleep for 5 or 10 or even 15 years, and then it comes back to kill the patient. Now obviously there's some component of resistance to dormancy, because if the drug had been effective, there wouldn't be any cancer to go to sleep and come back. So the resistance component to both of these. What we don't know is there a fundamental difference in the biology of early and late recurrences? And I'm going to get to that in a moment. For those of you who are not familiar, we have two basic types of ways of dealing with the estrogen receptor. The one we've had for longest is the approach taking anti-estrogens, where we compete with the endogenous estrogen for activating the receptor. That's why they're called anti-estrogens. And the newer class of drugs, they deprive the estrogen receptor of its ligand by blocking the ability of the body to convert androgens to estrogens. That's done with an enzyme inhibitor that blocks this enzyme called aromatase. So there's some similarity in both of these drugs in the sense that they're attempting to stop the estrogen receptor from functioning, but they do it in slightly different ways. So there is some level of cross-resistance between these treatments, but not complete cross-resistance. And the drugs are really surprisingly effective, despite what I said about the risks of recurrence. If you look here, for example, this is for tamoxifen-only treatment. This is the early breast cancer trial. It's cooperative group meta-analysis data. there's about a 45 to 50% reduction in the risk of recurrence and a 30% reduction in the risk of mortality. And that's at least as good as cytotoxic chemotherapy for postmenopausal breast cancer, without the toxicity, or many of the toxicities. And you can see here the cumulative benefit from tamoxifen. This again is old data where the only systemic therapy was tamoxifen. We couldn't do these studies today. You can see there's a significant overall benefit from these drugs. But still, some of the ones that respond will come back, and some that have the estrogen receptor won't respond at all. So when you look at this patterns of early and late recurrence, that led us to ask a very simple question. Are all recurrences on tamoxifen, irrespective of when they occur, are they the same, or is there some fundamental difference? Is there a difference between what we would think of as classical resistance and dormancy? And the way that we tried to ask that question was very simple. We said if they are different, we should be able to build robust molecular classifiers that can accurately separate whether a patient is likely to recur early or recur late. And if there's no difference, that would be an almost impossible task. And so that we did that by comparing very early, 3 years or less recurrences, with those that would recur subsequently later. So we have two data sets. The in-house data set came from our colleagues at the University of Edinburgh in Scotland. And we have 131 cases of estrogen receptor positive breast cancers. We have 15 to 20 years of clinical follow-up. And we have obtained the molecular profiles prior to treatment on all of these cases. And we looked and asked whether we could build a simple classifier comparing early versus late recurrence. And I won't go into all of the details of how we did this, but we used a fairly novel tenfold cross-validation approach using a support vector machine with the recursive feature elimination approach. One of the problems that we've recently discovered in breast cancer is that most of the molecular classifiers are no better than random genes. And even the 5 subgroups that have been defined and that are now considered absolute truth are not robust. There are a couple of wonderful studies, one out of the UK and one out of Belgium, that showed that none of these classifiers are robust. I can't explain to you why in 5 minutes, so I won't attempt to. But it has to do with the approaches that one takes to dealing with very high-dimensional data when you have large numbers of observations. So if you take one breast cancer and run it on something like an Affymetrix array, you get 40,000 variables for one specimen. And that's a relatively unique structure for data. And it requires a whole series of new approaches that largely have not been taken. This approach, using these types of tools, begins to address what's called the gene selection bias problem. That's a fundamental property of those high-dimensional data spaces. And the other thing is you need to have a very rigorous set of performance metrics for the classifiers. You'll see in a moment that we have 7 of those performance benchmarks that have to be met in both the training data set and in the independent data set. So to cut that story a little bit short, this is really the outcome. So this is comparing early, late tamoxifen recurrences. This is the in-house test set, the set from Scotland. These are our 7 performance metrics. And you can see all of them do 70% or better accuracy, which was our predetermined requirement. We had a little difficulty finding an independent data set because we had to get a data set that was on the same molecular platform with the same type of patients and with adequate long-term clinical follow-up, where tamoxifen was the only systemic therapy. But we found one, this data set from Loy et al., and we ran the classifier in exactly the same way on the same data set. And again, you can see a very consistent performance. They were all recurrent, that's why, or all died, and that's why in both cases you can see the survival curves come to the bottom. But you can also see there's a very significant difference between the two. So there's something that seems to be different between breast cancers that recur during treatment early, within 3 years, and those that recur 5 years or 10 years or 15 years later. So the next question is, can we begin to understand some of the factors that might be driving the failure of those endocrine therapies to be curative for these patients? And so we were forced a number of years ago to give up any hope of finding a single gene or a single mutation or a single pathway. It seems to be, particularly when you're looking at something that's driven by a transcription factor, somewhat more complicated than we would have liked to have considered. And that led us to take a more systems biology approach, assuming that we were looking at a gene network, not a signal transduction pathway, but a network. A network that is highly modular with both redundancy and degeneracy. So what that means is, the modules, there are different ways in which a cell can perform a single function. If you think about it simplistically, for alive or dead, a cell can die through apoptosis, or it can die through autophagy, or it can die through necrosis. Those are three different modules. That gives you redundancy. And then the signaling within each of these pathways also has the ability to deal with being altered. It's not always wired the same within each module, and that gives you degeneracy. And those two together give you a very complicated biology. So how do we begin to understand what are the most important functions? So for us, again, we took a very simple approach. We said the only things that we care about are the cancer cell is alive or dead. And if it's dead, we don't really care. And if it's alive, is it making copies of itself? Because if it's making copies of itself, it's going to kill the patient. But if it's sitting there for 20 or 30 or 40 years, that's not a bad thing. If a woman was diagnosed with her breast cancer at 60 years of age, and we put that tumor to sleep for 30 years, she's likely to die of something else. And I'd take that as a win. It might not be one way, the way you'd like to win, but I'd take it as a win. So we'd like to understand the alive or dead piece and the proliferation or not piece. So those are the two functions that we want to begin to understand. So I've shown you that there's a fundamental difference likely between early and late tamoxifen recurrences. They're all estrogen receptor positive. We know that if you take breast cancer cells and you make them resistant, whether you do it in the lab or it occurs in patients, they still tend to express the estrogen receptor. So there's something about the estrogen receptor that's still driving the biology of many of these breast cancers. And you can come along with a second line and a third line endocrine therapy and still get a good clinical response in many cases. So the first question that we asked next was, if the estrogen receptor is that important, can we learn something new about how the estrogen receptor might signal in breast cancers? We know it's a transcription factor. I've just shown you we have 2 data sets of transcriptome data. So what can we learn about signaling or about function by studying the data from those patients? The way that we chose to do that was we built a novel tool that does what's called Metropolis Sampling. It creates a random walk across the data and creates these short Markov chains. And the first thing we wanted to do was find out what in the known literature, in the known data, are the closest protein-protein interaction neighbors to the estrogen receptor. And to do that, we walked this algorithm exclusively across the 8 largest protein-protein interaction databases. We did short walks, only 2 steps per random initiation of the algorithm, but we did hundreds of thousands of iterations of the algorithm across each of the data sets. And that gave us almost 1,500 nearest neighbors. Then when you look at how frequently the algorithm walks from one protein-protein interaction to the next in those 2 short steps, you find that a relatively small number of proteins in those databases are actually selected. And you end up with somewhere around about 50 protein-protein interactions that are selected time and time and time again by the algorithm. So those are the ones that we identified as the initial nearest neighbors. Once we've got those 50, and we have the 2 data sets, we can do the same thing. We can ask, how are those 50 protein-protein interactions represented in the transcriptomes of early versus late recurrent breast cancers? And so that's pretty much what we did. The same data sets I've shown you, comparing the early and late distant recurrences with the 50 genes. And this gives us a very different view of just how the 50 closest protein-protein interactions for the estrogen receptor are likely to be represented in breast tumors. First of all, I don't have time to walk you through all 50, you'll see a few other genes in a moment, but apoptosis, that's alive or dead, more or less. The gene ontology in way which these are annotated in databases is a little loose. And cell proliferation, that's making copies of yourself. So the closest protein-protein interactions to the estrogen receptor are regulating the two functions that we think are the most important, but they don't do it in a particularly simple manner. What you see here is a representation of how those 50 are represented in early versus late breast cancers. And the size of the nodes of the genes tells you how frequently the algorithm visits it. And the thickness of the edges here tells you how frequently the algorithm walked from one to the other. Now, because these are known protein-protein interactions, you'll recognize a number of these genes and proteins. So we have BCL2, EGF receptor, of course the estrogen receptor, AKT, MAP kinase, and F-kappa B. But the issue is, if the signaling is as complicated as this, the cell has multiple ways when you knock out just EGF receptor or the estrogen receptor. It has multiple different ways of surviving and maintaining its signaling. And this is exactly what we see in the clinic. This would have predicted that the tyrosine kinase inhibitors, the PAN inhibitors that knock out EGF receptor family members wouldn't be particularly useful. And the initial studies out of most of the clinical trials in ER-positive breast cancers are showing they're not particularly useful. If you thought in a linear fashion, it would have made sense to target those, and that's why people did the experiments, started the clinical trials. When you start to understand that it's a network, not a single linear signal transduction pathway, you begin to see why cancer is so difficult to deal with when it comes back. So another way of trying to look into the system is to take cell lines. We can't do the functional studies in humans because it's unethical and technically extremely difficult anyway. So we have to go to cell lines. So you can take cells and make them resistant and ask what molecular features change when you go from sensitive to resistant, and are many of those represented in the clinical data sets. And we have no difficulty finding those, of course. And I was pleased to see the previous talk, because one of the ones we've been following for a number of years is actually nucleophosmin. In breast cancer, it turns out that that's an estrogen-regulated gene. And it actually interacts with another estrogen-regulated gene, interferon regulatory factor 1. It does so by binding and inhibiting it because this is a tumor suppressor gene. But I won't talk about NPM today. When you look at the types of genes that are differentially expressed, you find that a large number of them represent apoptosis, autophagy, and the unfolded protein response. And it's the unfolded protein response I'd like to talk a little bit more about today. So we have other evidence to implicate the UPR. This is a different way of looking into the data and asking the same question. This is taking cells that are sensitive, treating them with an anti-estrogen in a short period of time, and looking at the changes in gene expression that occur over time, and finding small topological features that represent the earliest changes in acquiring resistance. And you'll find that we see some of the same genes that are associated with acquired resistance. So early on, there are changes again in this XBP1 gene, which is one of the UPR genes. We have members of the BCL2 family, and we also see changes in NF-kappa B. So not only are cell lines reflecting tumors, but some of the earliest changes that you see in response to these drugs are maintained as cells acquire a resistant phenotype. And so the unfolded protein response, for those of you who don't remember their basic cell biology and biochemistry, when cells make proteins, they need to fold them. The folding of proteins is a very energy-dependent process. So if you're making a lot of proteins, you need a lot of ATP to fold them properly. The proteins don't get folded properly. They build up in the lumen of the endoplasmic reticulum that becomes stressed. And the cell activates the unfolded protein response to deal with that stress. Now, the UPR has 3 arms, and it uses them differently. It uses one arm to regulate cell survival, and to regulate the rate of transcription and translation. So one of the ways in which it deals with the stress is it tries to slow down the number of RNAs that are made, the amount of protein that's translated, so that the load on the endoplasmic reticulum goes down and the amount of energy that's available is sufficient to fold the proteins, and it can get back into homeostasis. To do that, of course, it has to survive, and it does that by the second arm, by sending out a series of signals that ultimately tell the cell to die yet because I've turned everything else down and I'm going to try and fix the problem that I have given the stress that's been applied to me. And that's what the UPR does. And we see a number of the members of the UPR differentially regulated in the acquired endocrine resistance phenotype. So you can map it. You can map all of the UPR across sensitive and resistant cells. What I'm going to focus a little bit on now here is XBP1. XBP1 came up in the human data. It came up in the cell lines that were made resistant, and it came up as one of the earliest changes, and it's one of the pro-survival pieces that comes out of the unfolded protein response. What happens is it's in two of the arms of the unfolded protein response. It gets differentially transcribed, gets turned on by ATF6, and it gets cleaved by IRE1 alpha. So it gets uniquely spliced, so you get this splice variant, and it's the spliced type form of XBP1 that drives survival. And you actually see the same pattern if you go into a rat mammary tumor model. So if you take Sprague-Dawley rats and you give them a chemical called DMBA, they get mammary tumors. Those mammary tumors are mostly estrogen receptor positive. You can treat them with tamoxifen. They will respond or regress. And if you look at the tumors that don't respond to tamoxifen, or that respond and come back, you see changes in the same signaling molecules. So we see it in patients, cell lines, and rats. So there's something fundamental about this unfolded protein response that seems to be able to drive that resistance phenotype. Other members of the UPR include NF-kappa B, and NF-kappa B is upregulated in resistant cells. It's no longer regulated by the estrogen receptor. If you come along with a natural compound that inhibits NF-kappa B, you can resensitize breast cancer cells that were resistant to the anti-estrogen. And the same with XBP1. If you take the mature spliced XBP1 and overexpress it in cells that are estrogen receptor positive, that require estrogen to grow, you can let them grow without estrogen. And if you take it into cells that require estrogen to grow and are anti-estrogen responsive, you can make them resistant to whether it's tamoxifen or fulvestrant or Faslodex. And that seems to be driven in part by the ability of XBP1 to regulate mitochondrial membrane permeability. So that's the pro-survival arm that comes out of the unfolded protein response that's activating NF-kappa B and XBP1 that seems to be able to protect the mitochondria. That's how the cell is deciding not to die yet. And it does that in part by regulating BCL2 family members. So you can see here in resistant cells, BCL2 is significantly upregulated. The most upstream regulator of the unfolded protein response that controls all three of those arms is called GRP78, glucose-regulated protein 78, and it's upregulated also. Now, if you looked at canonical signaling for the UPR, you would predict that BCL2 would be regulated by JNK and by CHOP. But in our models, that doesn't seem to be the case. In fact, if you remember the simple model with the little round dots and edges that showed XBP1 linking directly to BCL2, suggesting that rather than this sort of canonical signaling, BCL2 may be regulated by XBP1, which would give you redundancy within the system. So if you look in the promoter sequence of XBP1, there you find response elements, to BCL2, you find response elements to XBP1. If you overexpress XBP1, endogenous BCL2 goes up. If you knock down XBP1 in cells that endogenously overexpress it, down goes your BCL2. So now we have, from our models, predicted a direct relationship that's not a canonical signaling piece from the unfolded protein response, but that is providing a cell survival opportunity for the cells. And if you come along, because these cells that have acquired resistance now overexpress BCL2, which would lead you to think they may be more dependent upon BCL2 than sensitive cells. If you come along with a broad spectrum BCL2 inhibitor, the resistant cells are more sensitive. So that really leads us to think a little bit differently about how breast cancer cells respond to the stress of an endocrine therapy. No longer can we think in simple terms of a signal transduction pathway or a gene. There is an integrated, coordinated response that appears to be initially induced by the stress in sensitive cells. And as that response to the stress protects the cells, they learn how to maintain that stress response so that they can survive in the long term and become significantly resistant. As we've gone through this, I haven't had the time to show you all of the data because I want to pull it together in a way that tells you a sort of a more complete story. We recently looked at all of the data that we had that was in the literature to try and understand how we might begin to build mathematical and computational models of this signaling that would teach us something new about how the estrogen receptor signals. So I described that we think of this in terms of alive or dead or proliferating or not and modules. So what are the modules that would need to be regulated for cells to be able to survive the stress of an endocrine therapy? So in terms of thinking about individual modules, I've shown you that a significant amount of the early changes are driven by signaling that comes out of the unfolded protein response. So we've begun to think of that as the initial integration component because it controls the alive or dead, the cell survival piece. It does that, as you can see, through MMP, protecting the mitochondria through BCL2 family members. And at the same time, it also allows the cells to modify their response to the stress by changing the rate of transcription and translation through the other arms of the unfolded protein response. So what seems to be happening, and I don't have all the time to show you the data, but the upstream regulator of the unfolded protein response, glucose-regulated protein. When you treat with an anti-estrogen, you downregulate the glucose transporters, you downregulate the intracellular levels of glucose, you downregulate the intracellular concentrations of ATP. That's why the unfolded protein response gets activated. The cells no longer have enough energy to fold their proteins. That's how the initial stress of endocrine therapy is perceived. The pro-survival piece comes out of this signaling that includes genes like upregulating survival factors like XBP1 and NF-kappa B, and downregulating suppressor activities like IRF1. So we have two different levels of thinking about how this works. Things like programmed cell death or replication or growth are reasonably well evolutionary conserved functions. They may be wired slightly differently in breast cancer cells than they are in yeast or Drosophila, but they're not fundamentally different processes. What is different is what regulates them, what integrates the alive, dead, proliferate, or not component of the signaling. So while we're interested in modeling these pieces, there are actually quite good models for these already available. There's enough data to be able to adapt those models to the data that we have from breast cancer to allow us to build reasonable models for these, and we've recently built a mathematical model for this component of autophagy. The real piece is, can we understand the integration of signaling upstream that's likely to be relatively unique to the breast? So why does this make sense that the breast is important? It makes proteins normally during lactation. None of us would be here if the breast wasn't able to make proteins and do it for months at a time and survive and not die. So the breast is set up to use the unfolded protein response to sense stress responses, to deal with changes in its ability to make and fold proteins. It's not the only tissue that applies to. So here's where I think we really are. Early and late recurrence with tamoxifen, and it's probably also true for the aromatase inhibitors, are in some way different. Cells appear to adapt to the stress of endocrine therapies by integrating signals that regulate cell survival and metabolism. I didn't show you the metabolism data today, but you get a sense that if it's being driven by glucose-regulated proteins, that metabolism is going to be altered too. Yes, I would like to just finish for 1 second. The unfolded protein response probably is the initial sensor and stress response to endocrine therapy, and if it can get over that initial stress, the cell will use it to survive. It will use it to rebalance its energy metabolism, to survive, and to continue to grow. And all of this is really consistent with the way in which graph theory and network theory has begun to evolve in recent years. What it suggests is, and maybe it is unique to molecularly targeted therapies when you go after a transcription factor or a signaling molecule, rather than a genetic defect. Resistance probably doesn't require any new genes. We don't need to find new genes. We just need to understand how the genes that we already know about are wired and how they adapt to that stress. And so it's really the same network of nodes that allows cells to become resistant to these therapies. They just are wired differently. I will leave up, because I don't have time, the many people that helped us begin to understand this complex integration of responses to stress and signaling that we think allows some breast cancer cells to survive the endocrine therapies. You'll notice it's a very mixed group of people. We have biostatisticians, as you might imagine, but we have computer scientists and engineers who help us with many of those novel algorithms. The most important people I can't thank because I don't know their names, and many of them sadly aren't with us anymore. And those are the patients who provided the material for the clinical studies that I described at the beginning. Thank you.
I'm sure Professor Clark will be more than happy to answer a few questions from the floor.
Yeah. Can I go ahead with first? So when men have breast cancer, also can have the same classification, ER positive?
Yes.
And do they resist the same way as women?
I can't tell you that I know that they resist the same way. They get treated and managed the same way. They have the same molecular features: estrogen receptor, progesterone receptor, positive, negative, triple negative, or HER2 positive. You see that same distribution in male breast cancer. Probably it is largely the same. Because it's a relatively small proportion of all breast cancers, there are very few data sets that are large enough with male breast cancers that allow you to compare them directly to breast cancers in women. So for the moment, they get the prognosis is determined the same way and treatment is determined the same way.
Okay. Thank you, Dr. Clark. I was just wondering whether recurrences occur because of development of resistance with time or because of persistence of drug-resistant cells that were already there, which we now know as the cancer stem cells.
Yeah.
So is it more because of the persistence of those cells, or when development of resistance with time?
It's an almost unanswerable question. Both are almost certainly true. What we don't know is that the stem cells...
What comes first?
Sorry?
Which event occurs before?
I think it depends on the time frame of evolution of each individual cancer. So some of the estrogen receptor positive breast cancers are already resistant before they ever see the drug. And that's because something that they've inherited early on in their biology is already positioning them to be able to fail to respond. Whereas others seem to still have maintained that high dependence on the estrogen receptor when it's still present, and when you target it, they don't do well. So there's a time difference for the acquisition of those changes. Whether they would naturally occur anyway or not, I don't know. Whether the stem cells adapt very quickly to become resistant and then repopulate the tumor, or whether they're already resistant or not, I don't know. I don't know that anyone knows. And it's almost an unanswerable question in the sense that to change the signaling requires minutes. To acquire a new genetic mutation and have that expand requires months to years. So if you think about what's the simplest answer, if William of Ockham in the 11th century was more or less right, that what is the simplest answer is usually right, then the simplest answer is the cells have got everything they've got, they can adapt quickly, and it doesn't really matter whether it's a stem cell or another cell that does that adaptation. The adaptation arises and the women die. Best I can give.
I really enjoyed your talk. It's very comprehensive analysis of how network sort of poisons the system through adaptation. So can one use the modules of signaling networks and design chemical inhibitors that can combine with estrogens so that we prevent the learning process?
Yes, you can. We've already begun to develop inhibitors of XBP1 splicing. But what's particularly a little bit more encouraging, and it was hard to grasp from the complexity of the signaling that I showed you, but for some of the largest nodes in that network, there are already existing drugs. They're not necessarily used in breast cancer, but there are drugs to a number of those molecular targets, which would allow us to design some new trials with existing drugs that would give us an initial start. So there are two ways to look at it. There are what drugs do we have we can already use and repurpose? What drugs do we need to make because we don't have drugs to those targets?
Patient receives various types of chemotherapy like taxanes and other things. Will it have an impact on your nodal system or something to destroy the genes there so that the recurrences of the resistance developed for the estrogen receptors? Because when you see them at later dates, because all these patients undergo combination chemotherapies.
So, no, they are. The ones that we did, they didn't. They only ever had tamoxifen as a single therapy. That's why the system is clean. It's also why we'll never be able to do the study again. We do find the unfolded protein response components of it also being involved in drug resistance, which you might expect because there are other ways in which you can deplete energy and damage the cell, and part of it is, and endocrine therapies will do it also, they can change ROS production and damage DNA and activated DNA damage response, and that stress, part of that stress is also picked up by the unfolded protein response. So there's something about it that's a little bit more fundamental than I deliberately chose to.
Is the HER2 amplification has got any significance on this?
Yeah, I think if we could turn the early recurrences into late recurrences, for example, that would be good. Wouldn't be a cure, but it would buy a decade or two for women, and the opportunity to see your grandchildren, go to high school or whatever, and your daughter's married. That's a big win, even if at the end of the day you still get your breast cancer and you're going to die from it. And so I think we can learn how. I believe we can begin to learn how we can manipulate the biology to change it from a particularly bad outcome to a better outcome, even if we can't eliminate resistance. Resistance is tough.
Dr. Clark, so now that we know that PR is probably the target which you were specifically thinking of found out, is it possible to, how do you think we will be rewiring that target if you have to completely look it from a angle of gene therapy or something like that, which will give you a handle to bring down the resistance?
So the actual goal that we have that I alluded to but didn't talk about is you can take these data and you can build computational models that predict how the system functions. You can take all of the gene expression array data and how it changes over time. The models are usually built by a mix of ordinary differential equations and stochastic networks. But you can build a mathematical model with all of the data that you've got and see how the system works. The nice thing is you can then artificially change the values. So if you want to knock out the estrogen receptor, you can drive the value of that in your model to 0 and then see what the model predicts are the changes. And you can use the cell line or the rat models to test whether those changes are actually correct. So you can use computational models and mathematical models to learn how the system changes when you knock out the targets of interest. And if the models are adequate, they will never be perfect. But if they're adequate, they can at least tell you where the next big hit is that you need to knock out. So there are ways in which you can take these sorts of data and learn and try and do something new.